All right, I am going to finish chapter 29 talking about Protista. And I think we left off with the Apocomplexions. Okay, so Apocomplexions are called Apocomplexions because they have something called an apical complex. And you can see that in this diagram right here. And so the purpose of this apical complex is it allows them to invade a host because these guys are mostly parasites. They are spore-forming animal parasites. One type of apocomplexion that is well known is the one that causes malaria. And so they use their apical complex that has a bunch of fibrils, microtubules, vacuoles, well, some cell organelles at one end of the cell to get inside of their host. The apocomplexion responsible for causing malaria is called Plasmodium. There are a few other apocomplexions that can cause disease. Gregarines, which uses the apical complex to attach to intestinal epithelia of arthropods, annelids, and mollusks. And then we have the toxoplasma. You may have seen this term before, but it invades the epithelial cells of the human gut. And, you know, we can fight it off. We can mount an immune response to it. But pregnant women, you know, they always say avoid the kitty litter box because they could pick up this parasite from the cat litter, and that's the parasite responsible for that. So, you know, if you're pregnant, don't clean the litter box because if you should contract this parasite, it can really damage the baby's nervous system. Okay, so this is the life cycle of a plasmodium, and you can see that it's very complex. It's got sexual reproduction, it's got asexual reproduction, it has different hosts inside the mosquito, inside the mammal. So I guess we'll just start with step one. While feeding, the mosquito injects the plasmodium sporozoites into the human, and then it travels to the liver by bloodstream, gets inside the liver cell, where it will reproduce asexually and release these merozoites. Then these merozoites will multiply and get inside red blood cells that are released, and the cycle repeats itself. Certain merozoites develop into gametocytes, and then they can be ingested by previously uninfected mosquito and spread it again. But it hangs out in the saliva of the mosquito, where we have gametes that undergo fertilization, called an oocyst or oocyst to the sporozoite, and then just repeats. So efforts to eradicate malaria, we mostly focus on eliminating the mosquito vector. We also have developed drugs to poison parasites that have entered the body, and then just trying to develop a vaccine. Ciliates are another group in our aviolate. Wow. So remember, we've been talking about the aviolites, which were the dinoflagellates, the apocomplexions, and ciliates are the last group of the aviolites. So ciliates are named ciliates because they have a bunch of cilia on the outside arranged in longitudinal rows, or spirals around the cell. They can also have a pellicule, which is a tough but flexible outer covering. These guys do have two types of nucleuses. You can see that here in this purple and this pink color, the macronucleus and the micronucleus. Macronucleus is diploid. Without it, it reproduces asexually. The macronucleus is polyploid, so it's many copies of the DNA, and it's essential just to carry out functions for the organism. They also have two types of vacuoles. Food vacuole, which you can see here, okay? So it engulfs food and packages it into vacuoles. And then we have the contractile vacuole that helps with regulation of water balance, because these guys live in aquatic environments, and so water osmosis tends to rush in, so they package it in these vacuoles to kind of pump it out or bail it out. Continuing on with ciliates, conjugation can occur with the micronuclei. Only cells that are different genetically will conjugate together, and then meiosis will produce these haploid micronuclei, and then the two will exchange genetic information. And so let's see what step that is. Oh, I just realized the words are X'd out. I must not have copied over correctly. But step one, two different maiden types come into contact, and then divides to produce 4 new nuclei, and then makes the exchange. So here we have purple and pink, and you can see they have swapped some. So there's some pink over here and there's some purple over here. So, yeah. Okay, so within the group of the Chromoaviolata, there's 2 branches, the aviolites, and the second branch is called the Strematopiles. And there's going to be 3 groups that I'll talk about here, with brown algae, diatoms, and the Oomy Seeds, which are the water molds. So brown algae, large multicellular seaweed, aka kelp, very common along the temperate coastline because of the cooler water. They are not plants, and their life cycle does involve alternation of generation, which means they go from haploid to diploid. So we'll start right here with fertilization. Egg and sperm meet and fertilization, forming a diploid, and the diploid zygote will go through mitosis and develop a structure called the sporophyte. The sporophyte is the diploid, I don't want to say adult form, but it's the, I guess the mature form of the diploid side of things. And then the sporophyte will undergo meiosis and produce zoospores. And you can see that there are different sexes here. One's line for sperm, one line for egg. So here is the haploid stage. And then they'll meet in fertilization and start the whole process again. But the gametophyte is kind of like this mature haploid part of the life cycle. So that's brown algae. Moving on to diatoms, photosynthetic unicellular organisms. One thing that's very unique about them is they have this double shell, almost like a glass case that opens up, made of silica. They can also move along these things called rafes, these 2 long grooves lined with vibrating fibrils, as you can see out here. And then another thing unique about diatoms is that they produce a unique carbohydrate called crysalminarin. Okay. So going back to the glass case, you can kind of think of it as like 2 parts with that overlap. Maybe like a shoebox might be a shoebox with a lid. They reproduce asexually, with each daughter cell receiving half of the cell wall of its parent and regenerating a second half. So it's kind of like you take apart your shoebox and it will regenerate the other half. Okay, the third group underneath the Strematopiles is the Oomy Seeds, or water molds. They are either parasitic or saprobes, which means they feed on dead organic matter. Their spores are motile, which means they can move, spore or zoospores, and they move by flagella, so 2 unequal flagella on either end. Oomy Seeds, or water molds, are actually responsible for the Irish famine that occurred back in 1845 to 1847. About 400,000 people died from the Irish potato famine. And Oomy Seeds are kind of the leading hypothesis for the loss of fish and amphibians in ecosystems. All right, review questions. Which of the following characteristics supports molecular evidence for combining the dinoflagellates, Apocomplexions, and ciliates in the monophyletic clade Aviolata? The correct answer is E. Both or all 3 have a small membrane-bound alveoli under their cell surfaces. The organism that contributed to the Irish potato famine is C, an Oomy Seed. Oh, that's a typo. Mycete. A species that is photosynthetic with fine hairs on the flagella would be classified with which of the following groups? Stremnophilia. Next one. The organism that causes red tide is a blank and belongs to the clade blank. Red tides are caused by dinoflagellates and the clade Aviolata. In paramecium, removal of which of the following organelles would inhibit sexual reproduction? That was the micronucleus, the smaller one. More review questions. The disease malaria is caused by a species of the genus blank, which belongs to the blank. So malaria is an apocomplexion caused by Plasmodium. Which of the following organisms lacks chloroplast? D. None of the above lack chloroplast. They all have chloroplast. Final review slide. Which of the following produces a unique carbohydrate called chrysolaminiarian? That would be D, diatoms. And finally, the Stromatophilia include the... It is B, brown algae. All right, moving on to section 5, chloroplast and the Archaeoplastidia. So far we have talked about the excavata and this giant section, the chromal aviolata. So now we're moving on to this part right here. So I'll list the major characteristics of red algae, how humans use red algae, and then talk about how the Charophytes right here are closely related to the land plants. So a little bit of background. They acquired their chloroplast through primary endosymbiosis, but brown algae actually got their chloroplasts through secondary endosymbiosis. We know this, well, we've done extensive DNA comparisons with red algae, green algae, and brown algae, and it shows that red and green algae are actually closer together versus brown algae. Here's a picture of just some examples of archaeoplastids, very diverse. All right, red algae, Rhodophyta. They lack flagella and centrioles. They have accessory pigments belonging to the class Phycobilisomes. Phycobilisomes include phycourethrin, phycocyanin, and alleophycocyanin. But phycourethrin, I think of like red blood cells, okay? But they have accessory pigments, and that's why they're reddish in color. They can range in size from microscopic to 2 meters. And we actually use Rhodophyta or red algae in a lot of food, sushi wraps, ice cream, kind of like as a thickener, cosmetics as well. All right, chlorophytes and charophytes are green algae. So the green algae have 2 distinct lineages, the chlorophytes and the I don't know, I think I'm saying this wrong. We're going to call it the charophytes. Chlorophytes and the charophytes. So the chlorophytes, I highlighted in blue because, I mean, looking at these words, it's just, there's a lot of C's, CH's, they look very similar. So the chlorophytes are very, they kind of have like an unusual diversity. They kind of range in what they look like. They're microscopic, round, and green with 2 flagella. They live in the soil. They're haploid. They reproduce sexually as well as asexually, but they are not haploid diplonic. So they don't go through a haploid stage to a diploid stage and back. They don't spend some of their time as haploids and some of their time as diploids. The charophytes are the lineage that gave rise to the land plants, and we'll talk about evidence in the next couple slides here. So going back to the chlorophyta, cell specialization shows up in colonial chlorophyta. One example of that is the volvox. Volvoxes are basically this hollow sphere with a single layer of cells made up of either 500 to 60,000 individual cells. And each cell has 2 flagella, and a small number of them are reproductive, and that's where that specialization comes in. Not all of them reproduce, only a small number in that hollow sphere are responsible for reproduction. With the chlorophytae, there are some haploid diplonic life cycles, but then I'm going to introduce to you, I'm just going to throw another term out there, the streptophytes. Okay, the streptophytes. So we talked about red algae, okay, Rhodophyta. Here's my chlorophytes, kind of like my green algae. And now my charophytes belong to Streptophyta as well as my land plants. So the charophytes are more closely related to land plants, kind of gave rise to land plants. And we have molecular evidence to support that from rRNA and DNA, that they have haploid diplonic life cycles like plants do. Underneath the charophytes, you can subdivide it even further, which can be kind of confusing, between the charals and the coleochaetales, which is mostly freshwater algae. So I just made a table to kind of sort out the information your book gives you. So with the chorales, they're huge. They undergo mitosis and cytokinesis like land plants. But the thing that really sticks out is that they form green mats around ponds and marshes, and they just think, hey, it inched itself onto land. For the cholochatels, however you want to say it, they're microscopic, but they do have plasmodesmata, which is kind of unique in land plants. So because, land plants have a cell wall, they can't really like share information. Well, at least people didn't think that was the case, but then they noticed that they have these tunnels that connect each plant cell to each other, and they're called plasmodesmata. What they both have in common, they're both primary freshwater algae. Sexual reproduction relies on a large non-motile egg, so it doesn't move, but the sperm does. All right. Which feature of Charophytes provides evidence for them being the closest relatives to land plants? That would be A. Okay, moving on to the next section, Slender Pseudopods, which is Rhizaria. So here we are. We're going to talk about the Radiolaria, the Foraminifera, and the Circazoa. So talk about some distinguishing features between them, and we're going to talk about some slime molds. So the Radial Larynx, common marine phytoplankton. They also secrete an exoskeleton of silica, but they have these needle-like projections of pseudopods that it kind of stick out. Unicellular, and as you can see from this picture, they can be bilateral or radial symmetry. Foraminifera, marine phytoplankton. Instead of silica for their exoskeleton, they actually have a calcium carbonate shell called tests that leave this chalky deposit over time. And the white cliffs of Dover, that's why they're white. It's because of these Foraminifera. Size of them can be microscopic to a few centimeters in size. Mostly live in sand or attached to another organism. Kind of see up here what they look like. They do have apodia, thin cytoplasmic projections used for swimming, gathering materials for tests, their calcium carbonate shells, and feeding. Final group is called the Circazoa. They move with flagella or pseudopods. The silica exoskeleton is mostly, they have a silica exoskeleton, and they're mostly found in soils, is where I wanted to go with that. And one important piece with the Circazoa is that they're probably an organism that has recently undergone endosymbiosis compared to all the other groups that we've been looking at. So we're studying them to learn more about endosymbiosis. All right, review. The White Cliffs of Dover are made up largely of E. Foraminifera. Second question, in addition to the true amoebas, the Rhizopoda, another group with pseudopods are the C. Radial Larynx. Or Larynx. Excuse me. All right. Moving on to blunt pseudopods and amoebozoas. So talk about how amoebas move, and you can kind of see they're all by themselves here. So amoebas, they have these very, very thick or blunt pseudopods. And what pseudopods are, I'm not sure if we've defined it in this lecture or previous lecture or not, but they're projections of their cytoplasm. And they extend out, and they may attach to a surface and kind of pull themselves forward. They can also use these blunt projections to eat or engulf materials, as you can see right here. And there's microfilaments of actin and myosin on the inside that give that support and to help it move. Amoebas are most closely related to the Opisthoconta. A lot of them are free-living. They are beneficial to the soil ecosystem. They're a big part of it. But some are parasitic, and I'm sure you've heard stories of people contracting amoebas, and then it gets to their brain and it starts to eat their brain or something like that. Okay, another group, a part of the amoebas, is the Plasmodial slime molds. So this is what slime mold looks like. Okay, so they have a feed-in phase, and that's where they become visible. So they are macroscopically visible masses of oozing slime. And while they're in this state, they can undergo, where's this feed in? Here it is, feed in. They undergo multiple rounds of mitosis without cell division. So they have a whole bunch of cells that have a lot of nucleus on the inside. Another cool thing about plasmodial slime molds is that they can migrate if the food or moisture is in short supply through spores. So they'll form this stalk, and then a structure called the sporangium, which stores the spores. The spores will undergo meiosis and then be released. They are flaginated, so hopefully they find a new place to live and then undergo syngamy to form a diploid and start the whole process again. There's another type of slime mold, and that is the cellular slime mold. So the cellular slime mold, one cool thing about them is that we think that cellular slime molds were kind of the first multicellular examples of organisms exhibiting multicellular features, or just being multicellular in general. But they do have a relatively simple developmental system, and we tend to study their cell differentiation. They can also talk and signal or interact with other cells. And these cells, because they communicate and interact with one another, it's an aggregate organism. So we have cells that are somewhat individually, but they're communicating and working together with other cells. And so when this happens, we call that actually a slug. Okay, I think I have... Yep, this is a repeat question, sorry. So we'll just skip this. It was the Oomy seed. One of the earliest examples of multicellularity occurs in that would be D, cellular slime molds. Next review question. When food is scarce, cells of this organism communicate with each other to form a multicellular slug. That would be A, cellular slime molds. Okay. The final section. Propulsion via a single flagellum in the Opisthoconta. So we are finally approaching the last supergroup, okay, the Opisthoconta, which we're going to talk about the cyanoflagellates. They are grouped with the fungi and animals, but these guys are protists, and this is fungi, and these are animals. So we'll just talk about the cyanoflagellates here. So why are they so important? Well, these guys are unicellular organisms, and they are very similar structurally to freshwater sponges. Okay, so I know maybe you can't see in this picture here, but this is a sponge. In sponge, they filter feed. Okay, they bring materials in, and then there's these flagella that move. Actually, it's probably the other way. They bring material in, and it comes out this way. I think that's the way it goes. But these flagella move, and they create tiny little currents, and they filter feed. So they move these flagellas, and they filter feed, and then brings the food in, and expels water and other materials out. Now, cyanoflagellates, you can see they are very similar. I know this is a cartoon drawing, but here's a real picture of it, and you can see the flagella kind of moving here. So this single element flagella is surrounded by this funnel-shaped contractile collar. And so, yeah, they just, this structure is similar to what you can find in sponges. That's one piece of evidence, that shows that cyanoflagellates are closely related to animals. Another, and this is the new actually, they have found a homology of a surface receptor in sponges that is responsible for phosphorylation. So just more evidence that cyanoflagellates are probably ancestral to animals, aka sponges. Yes, sponges are animals. All right, final review question. Which is most likely the ancestor of animals? That would be D, cyanoflagellates. Okay, so that does it for Protista. And yeah, we'll carry on with seedless plants.